Hi students, today we are going to discuss about the carbohydrate metabolism important short notes, galactose, overview of galactose metabolism, right? You all know galactose is one of the monosaccharide, very important short notes, right? You have actually routinely they are asking clinical scenario galactossemia only. Before that, you should know about the galactose metabolism. Then only it will be easy for your clinical scenario or metabolism like questions. Okay. So for this galactose metabolism, I am going to tell some 8 points only. Very important 8 points. How to draw the metabolism in a simple way, right? So first step is galactose is converted into galactose 1-phosphate. Here the enzyme is galactokinase. Kinase means the reaction will be utilizing one molecule of ATP which is converted into ADP. This is the first step. This galactose 1-phosphate combined with one molecule of UDP glucose, right? So for galactose metabolism we need one molecule of UDP glucose, that is uridyl diphosphate glucose. So combination of these two will produce another two products, that is glucose 1-phosphate and UDP galactose. Very important step. Here galactose 1-phosphate and uridyl diphosphate glucose which is converted into glucose 1-phosphate and UDP galactose. This is the second step. Here the enzyme is galacto 1-phosphate uridyl transferase, otherwise known as GALT, G-A-L-T. GAL means galactose 1-phosphate, T means uridyl transferase. Galactose 1-phosphate uridyl transferase is the enzyme for the second step. You should know about these two very important enzymes. What is the enzyme disease involved in this step? What is the disease involved in this step? That is why you should memorize these two enzymes in galactose metabolism, right? Third one is, now we are having two products in our hand. This one is glucose 1-phosphate. This will be converted into glucose 6-phosphate by phosphoglucomutase. Mutase means 1-phosphate to 6-phosphate. Finally, this glucose 6-phosphate, depending upon the concentration of the glucose, either it will be converted into glucose molecule directly or sometimes it will go to the glycolytic pathway. So what is the fate of this glucose 6-phosphate in galactose metabolism? Depending upon the glucose concentration or energy, this glucose 1-phosphate directly converted into glucose and gives the energy, or otherwise they will enter into the glycolysis and produces ATP like energy molecules. Second product is UDP galactose. What will be the fate of this? Two fate of this galactose. One is this UDP galactose is converted into again UDP glucose by epimerase. Epimerase means one monosaccharide converted into another mono, that is UDP galactose will be converted into UDP glucose with the help of epimerase, and this UDP glucose, depending upon the concentration, they will go and get deposited in, converted into glycogen. Okay, so here the UDP galactose converted into two products, that is glycoprotein, glycolipid, GAGs. So for the synthesis of GAGs, glycolipid, glycoprotein, UDP galactose is very important. And depending upon the situation, they will be producing lactose, another disaccharide. This is actually monosaccharide which will be converted into lactose with the help of lactosynthase, right? So these are all the steps involved in galactose metabolism. Okay. The last one is clinical important question. Suppose the galactose, actually if the GALT enzyme deficiency produces accumulation of galactose 1-phosphate, that is why I am separately giving accumulated product for galactokinase and a GALT, that is uridyl transferase. Galactose 1-phosphate uridyl transferase deficiency will lead to the accumulation of galactose 1-phosphate, important MCQ, whereas galactokinase enzyme deficiency will produces accumulation of galactose. This galactose will be converted into galactitol in the lens and retina, and they will go for the formation of cataract, right? Congenital cataract. Next one is the accumulated product and galactitol formation. Finally, usually they will be asking your clinical scenario in this topic is galactosemia. In that galactosemia, these are all the subtitles. That is, you have to write 1 or 2 points for the introduction, then coming to the name of the enzyme defect, and then you have to mention what are all the problems or clinical manifestations of that babies, then what are all the investigations you want to go for, and finally how will you manage the galactosemia, right?